did respond better than the mice colonized with the microbiome of non-responders. And as you can see on the right, there was also a link again with Faecalibacterium. So now the question is, basically, what can we do for this patient non-responding to immune checkpoint inhibitors? So in 2021, these fecal microbiota transplantation studies were published, basically back in Science, and these studies were open label, so no control group, but still, these two studies showed that FMT with microbiota from responders were able to rescue the efficacy of immune checkpoint inhibitors in some patients who lost the response. You can see on the left that clinical response was observed in 3 out of 10, and on the right in 6 out of 50. And actually, one of the signals here was also the enrichment in Faecalibacterium positivity following FMT in patients who responded to the fecal microbiota transplantation treatment, really again suggesting some link with this bacteria. So now, can we really just test directly whether Faecalibacterium positivity is able to improve response to immune checkpoint inhibitors? So actually, we can do that, and we did. So we used the strain developed with Excellium because obviously this strain was already in clinical development for IBD, so it was relatively easy to use the same drug for immuno-oncology. So we first used it in mouse models. We used a very conventional model, sarcoma, MCA205, tumor-bearing mice model. We induced some kind of alteration of the microbiome to decrease the efficacy of immune checkpoint inhibitors. So the idea was not to wipe out the microbiome, but really just to induce some kind of perturbation. And then the mice were treated with anti-PD-L1, with or without Faecalibacterium progeny EXL01. So what you can see here on the tumor volume is that anti-PD-L1 is working. In red, you can see that the curve, the tumor growth is much slower than in the vehicle group, but you can also see that antibiotics impair the response to immune checkpoint inhibitors. And interestingly, when on top of antibiotics we added EXL01, actually we rescue the efficacy, and this was even clearer when we looked at the mortality curve. You can see that with antibiotics we basically lose the efficacy of anti-PD-L1 and we completely rescue it with the EXL01. So we did some RNA-seq analysis of the tumor, on the tumor, and what we see is that first, when we compared mice treated with anti-PD-L1 to mice not treated with anything, obviously almost 4,000 genes were differentially expressed, so of course something is happening. When we look at the effect of the antibiotics, again, several thousand of genes were differentially expressed. Then the effect of EXL01, again, more than 4,000 genes were differentially expressed, so really something is happening in the tumor. And interestingly, when we compare the mice treated with anti-PD-L1 alone to those who receive antibiotics, anti-PD-L1 and EXL01, and now we have a very small number of genes that are differentially expressed, suggesting that the EXL01 is able to basically counteract the effect of the microbiome perturbation.